Ba boom. This is the pea glow plasmid inserted into some E. coli. So a lot of this past work, I've been purifying the plasmid, and then I finally did this heat shock treatment that opens up pores in the bacterial membrane and then allows this plasmid that I purified to get inside, and then it starts transcribing and translating into proteins that fluoresce under blue light. And that's super exciting because 2 days ago, or well, I plated them 3 days ago, but then I came in 2 days ago and I saw basically no growth and no glowing. And I was like super bummed. I thought it didn't work. So I was like, all right, I got to redo this whole thing. And I probably still will redo it, but it's really exciting to see these colonies. But one thing that got me thinking, I was like, why did this take so long to start growing? And it got me thinking about bacterial growth and particularly the stages of bacterial growth. There are four main types. Stages. There's the lag phase in which the bacteria are sort of getting accustomed to the environment. Then there's the exponential phase where they're actually doubling and they're growing. And then the stationary phase is where the food is sort of exhausted and they're sort of settling down into less growth, no growth. And then finally, they've exhausted all the food and then they're dying off. So I wanted to see, can I recreate this plot? Can I basically take some media, start culturing it, and then sample the basically concentration of cells in my liquid media at different time points and see, is this really what I see in reality? Is it not just a model? And so I set out to do this at lab. Basically, I made a bunch of media. I then, poured them into some 50 milliliter conical tubes. Is not really relevant. But then on one of the tubes, one of the 50 mils, I inoculated it with a colony. And we'll get back to maybe why I did a colony. I didn't have any liquid media on me, but this is actually probably a big source of error and why this lab didn't go exactly how I wanted. Let me actually just put up the plot. So this is the data that I have, and we can see here that the 37 eventually surpasses the 30. So these are temperatures. Basically, I had three environmental conditions. One, I just left the culture tube out in room temperature. Then I put it in a pretty fast shaking, I don't know the RPM, 30C shaking incubator. Then I put it in a pretty slow shaking, I think 100 RPM, 37 degree. And I was really hoping, at least at the bare minimum, to see that the 37 grew the fastest. And that indeed ended up being the case. And I still don't really like this data. And I think just to get back to the auger culture, is that taking a colony from that, I didn't vortex it enough so that the media that I was starting out with when I poured my initial culture tubes that gets separated out, I didn't actually have basically enough homogenization of it. So it's likely that there was like more cells in one of the culture tubes than the other just based on how I poured it. So what people typically do in these growth curve experiments is that they back dilute already grown liquid culture. And this is going to bring me to the thesis of this whole day, which is I really hate how tedious and manual this whole thing was. I mean, first off, I was surprised that it even took 5 hours to start getting into the log phase, 4 or 5 hours to get into the log phase, even at 37C. That was pretty surprising to me. I think it would be interesting to model that more detail, in more detail, including like all the metabolism that's going on and everything. But anyways, this got me thinking about, this is one of these things that I bet biologists have to do all the time, just a lab where they're babysitting this stuff. They're taking a let me explain the experimental setup because I feel like that's important context. So we have basically three 14 milliliter culture tubes in different environments, the room temperature, the 30C, and then the 37C. And basically every 30 minutes, I would go collect my culture tubes. I would then take these cuvettes that I had, and I would add 1 milliliter from each environmental condition conical culture tube into the blank cuvette, and then I would blank it using at the very beginning of this, I took 1 mil of LB that I started with, and I put it into a cuvette, and then every time I took a sample, I blanked on that. And I think that this also could have been a source of error because if there was contamination in, which honestly I don't think there was, because I autoclaved the LB, but the thing is, it was sitting out exposed to the air, so something could have landed on it and started to grow. And there is some evidence that this may have happened in the plot. Let me put the plot up again. So while the room temperature culture was not exposed to air, but the blank cuvette was, the fact that the blue line is so flat maybe indicates that we're, I don't know. There's a number of ways to interpret this. Because we get to an exponential, my guess is that the blank actually never got contaminated, but it is probably something that I would do differently. But in general, what I was thinking of doing that would be really cool is to use the lab's OT2, OpenTron's robot, and maybe try to jerry rig it so that I could get 3 heat blocks in there at different temperatures, and we wouldn't have shaking, but that's a nicety that I mean, the whole point of this is to do an experiment where we're controlling for a single variable, which is just doing basic science. And what we would do then is we'd be able to, somehow take the heat block and fill like 1 milliliter of culture into, we'd have our culture, and we'd inoculate it with the original starting liquid culture. Then we'd have the robot basically add 1 milliliter to a bunch of tubes, and then we would basically every so often they would be in the heat block, and it would just take one of those pipettes or one of those like 1.5 milliliter Eppendorf tubes, take a milliliter from that and put it into a cuvette that's in some custom labware that I designed. And then basically it would, it would run I don't know if there's a way to give input into the OpenTrons, but basically what I would like to do is it would fill the cuvettes. I would take the cuvettes manually over to the spectrophotometer. I would record my data, write it down, and then I would return the cuvettes to where they need to go. And then the machine would remove the media into a waste container, and then it would also refill it 30 minutes later, so that then I don't have to babysit as much, because I spent 6 hours, like, it doesn't feel like a waste, but it was just like, I couldn't do anything in the interim. Like I had all these plans to, like, okay, I'm going to make more competent cells, I'm going to do all this, but I just didn't have time because I just felt rushed or whatever. So this is one of these things where it's like just suboptimal use of my time because I'm babysitting this experiment. So I've been thinking about how we can improve that. All right, so most of the rest of this footage is all just, sampling, doing the same thing over and over again. However, towards the end, one thing that's really cool is I joined in on this project to make real vegan cheese. And I don't really know anything about cheese, and I don't really know about this project. But from what I gather, the goal is to produce a couple of the main proteins that are in cheese in yeast. And now the thing that's really interesting here is that yeast is eukaryotic. And if I understand correctly, I really need to just get back to doing some reading and like learning, because this is learning, but it's not theory. And basically we need to incorporate the gene for, I guess it's called beta casein, into yeast. And the interesting thing about this is, well, it's not just putting a plasmid in. It's more difficult than that. So I don't know how we do that. I need to read about that. But basically what we did, a couple of other college students came, and actually I got my first, this is crazy to me. I have 500 subscribers and still somebody was like, "Oh, I saw your YouTube video." That's never happened to me before. I was geeked, man. That was sick. Anyways, but basically they had some data of, like, supposedly they had incorporated this beta casein gene into this yeast, and there were these frozen samples. They thawed them, and then we looked at some of the spectrophotometry data to see if there was a noticeable increase or change at a given wavelength to indicate that beta casein was actually being produced. And the data was like super inconclusive. And what we sort of decided on is that maybe it would be a good idea if we tried to just first start more simple and add a gene for some sort of colored protein. There's some special name for colored proteins into yeast, so that we can see visibly whether or not the protein is being produced. Because it's really easy to just look at the test tube and be like, "Oh, it's blue." well, then the blue protein is obviously being produced because none of the other ones are blue, or the wild type is not blue. By the way, don't like the name wild type, but I don't know. It's just confusing to me. But now I'm sort of getting used to it. Anyways, the point though is how to basically we need to go in two stages because we have this blue plasmid, I think. But the trick is that we need to get it first into E. coli so that we have it propagated. Because once, and the interesting thing about this is this requires a new technique because it requires, I think it's called Gibson assembly, but it basically is a technique to insert a gene into a plasmid. And that, I hadn't even thought of that before because I've just been given the plasmid. But what happens if you need to make a plasmid? Well, you need to, anyway, so it's a super powerful technique, and I need to learn a lot more about it and how we're going to do this. But basically, the goal is take this, and I don't even know how you take a gene and put it into a plasmid, really, because it's like, yeah, I don't even know how we're going to do that. But somehow, we're going to basically take the blue gene. I don't know if this is right. Actually, on the whole, I know we're at the end of the video, but if anybody has like advice or like just shout me out for doing something wrong, I really want to know that because I feel like a lot of this is that I do is sloppy. But anyways, we're going to try to get this blue protein first into a plasmid, then we're going to express it in some E. coli, and then finally we should be able to do this process where we can cut out the gene and incorporate it into the yeast genome. And so we're going to try to go from there, and I'd love to pick up this project because honestly, repressilator is seeming a less and less fascinating the more, just because it's like, it's the same thing. It's just a transformation.